Welcome to the Molecular Biology Overview, brought to you by the SMART team at Washington University in St. Louis. This short lecture series aims to provide a concise introduction to the fundamental principles of molecular biology. Molecular biology centers around the central dogma, which explains how genetic information flows from DNA to RNA and ultimately to proteins. This process involves transcription, where DNA is transcribed into messenger RNA, and translation, where the RNA sequence is decoded into a protein. From a metabolic engineering perspective, enzymes play a crucial role. They catalyze biochemical reactions, driving metabolic flux toward desired final products. This understanding bridges molecular biology with practical applications in metabolic pathway design. The DNA, or deoxyribonucleic acid, is the molecule that stores genetic information. Structurally, it has a sugar phosphate backbone with nitrogenous bases. The two helix are held together by hydrogen bonds. DNA's double helix structure consists of two strands running in opposite directions, one 5' to 3' and the other 3' to 5'. This orientation is crucial for biological processes like replication and transcription. Chemically, each nucleotide contains a phosphate group, a sugar, and a nitrogenous base. RNA, or ribonucleic acid, is another key molecule in the flow of genetic information. Unlike DNA, RNA contains the nitrogenous base uracil instead of thymine pairing with adenine. Its bases include adenine, uracil, cytosine, and guanine. There are several types of RNA with distinct roles. Messenger RNA, or mRNA, carries genetic instructions from DNA to ribosomes for protein synthesis. Transfer RNA, or tRNA, helps in assembling amino acids during translation. These diverse RNA types collectively ensure the accurate expression of genes. Polymerase chain reaction, or PCR, is a powerful molecular biology technique used to amplify specific DNA sequences. The process relies on several key components. First, the template DNA serves as the blueprint containing the sequence to be amplified. Short single-stranded primers are added, which are designed to bind to specific regions on the DNA. DNA polymerase extends these primers by adding nucleotides, the building blocks of DNA. The reaction is carried out in a buffer solution that provides ions and ensures optimal pH for the enzyme's activity. A PCR involves three major steps that repeat in cycles. The first step, denaturation, occurs at a high temperature of 94 to 98 degrees C, where the double-stranded DNA separates into single strands. Next is annealing, typically performed at 50 to 65 degrees C, where the primers bind to complementary sequences on the single-stranded DNA. Finally, in the extension step at 72 degrees C, DNA polymerase synthesizes new DNA strands by adding nucleotides to the primers. These steps are repeated multiple times, resulting in exponential amplification of the target DNA sequence. Synthetic biology systems are designed using standardized genetic components that work together to create a functional biological system. These components include promoters, ribosomal binding sites, open reading frames, and other regulatory elements. At the core of these systems is the expression cassette, which consists of three main parts. The promoter serves as a binding site for RNA polymerase and can be regulated by activators or repressors. The ribosomal binding site, or RBS, facilitates the attachment of ribosomes to the messenger RNA, ensuring proper translation. Finally, the open reading frame, or ORF, contains the coding sequence starting with the start codon ATG and ending with a stop codon like TAA. These modular parts can be engineered and combined to achieve precise control over gene expression, enabling synthetic biology to tackle complex challenges. Promoters play a central role in gene expression by serving as binding sites for RNA polymerase, the enzyme responsible for initiating transcription. In this example, the PTET promoter illustrates how transcription can be regulated by repressors. In the absence of a repressor, the promoter is active, allowing RNA polymerase to bind and initiate transcription of the downstream gene. This is the on state. However, when the repressor protein, TET-R, binds to specific operator sites on the promoter, referred to as O1 and O2. It physically blocks RNA polymerase from accessing the promoter, turning transcription off. Regulation can be reversed with an inducer like tetracycline. When tetracycline molecules bind to TET-R, they cause a conformational change that releases the repressor from the operator sites. This release restores RNA polymerase's access to the promoter, switching transcription back on. This inducible system allows for precise control of gene expression in response to external signals. The PBAD promoter demonstrates how activators regulate gene expression. In the on state, the presence of arabinose creates an Ara-C-arabinose complex. This complex binds to activation sites, allowing RA polymerase to attach to the promoter and initiate transcription of the downstream gene. In the absence of arabinose, the system switches off. Here, free RSC proteins bind to operator sites O1 and O2, forming a DNA loop. This loop physically prevents RNA polymerase from binding to the promoter, effectively inhibiting transcription. This dual functionality of RSC provides tight regulation of gene expression, making the PBAD promoter highly responsive to environmental conditions. Identifying genetic parts is a fundamental step in synthetic biology and molecular biology research. One of the primary resources for this is the NCBI's Annotated Gene Database, which provides detailed information on gene sequences, their functions, and associated biological pathways. When working with DNA sequence data alone, genes can be identified by locating start and stop codons, as well as ribosomal binding sites. Tools like BLAST allow researchers to compare DNA sequences to known databases, while reference genomes are invaluable for analyzing RNA sequencing. Together, these resources streamline the process of finding, annotating, and utilizing genetic parts for research and engineering. Enzymes are essential biological catalysts that accelerate metabolic reactions by lowering the activation energy required for these processes. They play a pivotal role in nearly every biochemical pathway, ensuring that reactions occur efficiently and at the right time. Enzyme engineering is an active area of research focused on modifying enzymes to improve their functionality, specificity, or stability for industrial and medical applications. However, precise engineering of enzymes remains a significant challenge. This is due to the complexity of enzyme substrate interactions and the intricate three-dimensional structure of proteins. For example, the study referenced here demonstrates how substrate inhibition can be abolished through protein engineering, improving the productivity of lycopene synthesis. Despite advancements, understanding and manipulating enzyme dynamics continue to require cutting-edge techniques and tools. Metabolic reactions within a cell form an interconnected network, commonly referred to as metabolic pathways. These pathways map out how different molecules are synthesized, broken down, and transformed to sustain cellular function. A valuable tool for studying these reactions is the KEGG database, which provides a comprehensive resource for visualizing and analyzing metabolic pathways. For example, pathways such as glycolysis or the citric acid cycle can be explored in detail with information on specific enzymes and intermediates. By using tools like KEGG, researchers can analyze metabolic flux, the flow of metabolites through these pathways. This insight is crucial for applications like metabolic engineering, where optimizing flux can lead to higher yields of desired products or enhanced metabolic efficiency. Metabolic reactions and flux can also be studied using genome scale models, which provide a system-level understanding of an organism's metabolism. These models integrate various types of data to map out all potential reactions within a cell. Constructing genome scale model involves several steps. First, genome annotation data is used to identify gene functions and their presence in the organism. Next, reaction data is added to define the compounds and stoichiometry of each reaction. Finally, a constraint-based modeling is applied to incorporate physical and chemical constraints such as mass balance and thermodynamics, ensuring accurate predictions of metabolic behavior. The BIGG database is a powerful resource for exploring genome scale model. It allows researchers to search for models, reactions, metabolites, and genes. Genome scale models have various applications in understanding and manipulating metabolic networks. One major application is studying cellular resource allocation to optimize biological processes. Genome scale model is also important in designing strains with desired flux phenotypes. However, genome scale models come with certain limitations. They often oversimplify complex biological systems, which can lead to inaccurate predictions in real world scenarios. Additionally, constructing a reliable genome scale model requires high quality data to constrain and validate the model, making the process both data intensive and computationally challenging. Despite these challenges, the genome scale models remain a vital tool in systems biology and metabolic engineering. The gold standard for measuring metallic flux is E13C metabolic flux analysis. This technique provides precise insights into the flow of metabolites through cellular pathways by leveraging isotopic labeling. There are two approaches to 13C based studies: qualitative 13C fingerprinting and quantitative 13C metabolic flux analysis. And 13C fingerprinting is used to investigate functional pathways without performing detailed flux calculations. It relies on analyzing isotopomer data from targeted 13C labeling experiments, offering a simpler, less computationally intensive method. On the other hand, the 13C metabolic flux analysis uses nonlinear least squares regression modeling to resolve fluxes within the central carbon metabolism. It accounts for stoichiometric and isotopomer balances to provide quantitative data on metabolic pathways, offering a more comprehensive understanding of flux distributions. Performing 13C based metabolism analysis involves several critical steps to quantify and analyze metabolic fluxes. Step 1 begins with building a metabolic network, which defines the pathways and reactions involved in cellular metabolism. In step 2, growth rates and extracellular exchange rates are measured. These include the uptake of glucose and the production of biomass and byproducts. Step 3 involves extracting amino acids from the biomass. These amino acids carry isotopic labels. Step 4, derivatization, chemically modifies the amino acids to make them volatile and suitable for downstream analysis. In step 5, the samples are analyzed using gas chromatography, mass spectrometry, or GC-MS. This technique identifies isotopomer distributions, providing data for flux calculations. Finally, step 6 uses computational tools to calculate fluxes within the metabolic network. By integrating experimental data, researchers can model the flow of carbon through metabolic pathways and gain insights into cellular behavior. Thank you for listening to this presentation. I hope this overview has provided valuable insights into molecular biology, synthetic biology, and metabolic flux analysis. If you have any questions or wish to dive deeper into any topic, feel free to reach out. Thanks again for your attention.